Welcome to a mini-series of Neurosalience, featuring interviews with some of this year's annual meeting's keynote speakers.
Well, let's get started. Thank you again for interviewing. So, Alessandro, can you start by describing a bit about your background and what you study, what you research?
Yes, sure. Yeah, thanks for having me. It's a great pleasure to have the chat and share some of the stuff we've been doing in the informal discussion. So, briefly, I lead a research lab in the north of Italy. My institute is a tech institute, so a research only institute. It's called Italian Institute of Technology. My lab is called Functional Neuroimaging Lab, and a big focus of our work is to bridge the gap between experimental neuroscience, which is something that you tend to do a lot in animals, so like this more basic neuroscience, and human brain map. And this is something that we embarked upon like a few years ago. To do that, to achieve these goals, actually, we have pioneered the cross-species approaches and perturbational methods to try and establish causality between events at different scales. And we have clearly a strong focus in translational imaging, so the application of imaging approaches such as fMRI, for example, both to animals and humans, as a way to, as I said, bridge these different fields.
So there's a lot of different aspects to this that you mentioned. So there's cross-species translation. You do, from my impression, a lot of engineering work to go with that. So it's a very interesting and interdisciplinary background, I would say, that you have. Can you speak a bit about how you got to this stage, what your training background was?
Yeah, sure, definitely. And yeah, I mean, my background is for sure unconventional because I originally trained as a molecular biologist. And then I started out in the pharmaceutical industry where I worked for 9 years or so. Well, before that, actually, I kind of found my way accidentally into brain imaging. So I shifted. And then, I ended up in academia. We don't really want it to be here. And of course, I love every minute of it. So this was really like unplanned and unconventional. I'm very happy to summarize, if you like, yeah, my path, because I think it holds maybe a lesson also or two to young generations. So basically, I grew up like in a small like farming village, and I wanted to become a farmer. That's where it all started because that was the word I knew. And so I began to study agriculture. But then I realized at a certain point that, if you want to be a farmer, you need to own a farm, right? And I didn't. So, I mean, I realized it took me surprisingly long to figure that out. So I retrained in molecular biology because that was like late 90s. So there was like the biotech era. There was, human DNA mapping. So there was a lot of hype about molecular biology. And of course, I had always been like interested in science and technology. So I thought, okay, I could reprogram myself into that. And I got a degree, a master's degree in molecular biology and bioinformatics. And as part of a master's thesis, I was hired by a company, like one of these like research-based pharmaceutical companies. And I worked for a couple of years as a like molecular biologist, bioinformatician. And then a pivotal moment happened at first. There were really two big pivotal moments in my career. The first one was basically that the branch I was working for was basically shut down as part of these mergers. These big companies, they merge all the time, they chase each other. And so basically, I had been made redundant and, I was out of a job. But I was also given the chance to apply to other positions. And that's really how I found my way into imaging because the same company on the same site was like setting up one of the first preclinical scanners, MRI scanner for animals. I found that really fascinating. That was also the time where, we were also at the dawn of the functional neuroimaging era in humans. So, I was really intrigued by that. And I applied and I got hired there. And that's when we had the vision that we could like implement drug mapping in the rodent brain to do a bit of a fingerprinting, which is something that people are doing now also in humans. That was like 20 years ago. And we started publishing work, because it was innovative. And I began to be interested in the idea of like publishing the academic system. And so I decided to get a PhD while working, right? So I was working, and in my spare time, I used like some of the data I had to get a PhD at a local university. And then the second pivotal moment came, which was again, restructuring, a new merger. And again, the branch I was working for was shut down. So I was again made redundant. But in that case, I was kind of ready because had been like applying for academia because I wanted more freedom. And so here I am. Yeah, that was like 2010. And then, I was lucky enough to get my current job at the Italian Institute of Technology. So yeah, this is really like an interesting story, at least with the eyes of my students, because I've been very shy about this for many years, right? Because clearly I don't have like the academic pedigree one would expect me to have. But my students find this story kind of liberating because it's interesting that life has got way more imagination than we do, right? And so life, I ended up being there out of necessity, right? More than planning. And then I am exactly where I wanted to be. So,
Well, it's a very reassuring story, I think. I know that at least a lot of people in my circles, a lot of trainees are trying to find the balance of academia and industry. And, we often hear things like it's very difficult to get into academia after going for industry. And I think this is a great example of you breaking down that barrier. So it's very nice to hear. Okay, so I'm going to switch tracks a bit. So in your description of research, you talked a bit about cross species. And it's interesting because in the past few years in OHBM, we've been seeing a lot more preclinical non-human research being featured. So I think in a couple of years ago, the Tolerate lecture was about the mouse. I think almost every keynote lecture in last year's conference in Korea featured some preclinical component. And so I'm just curious, what does preclinical imaging bring to the table, in your opinion?
yeah, sure. I mean, let me just start by saying that I'm super pleased about this trend that I've seen happening because for many years, preclinical and experimental neuroscience was in its own silo and fMRI was in its own silence. And these were like separate, right? And there have been many advocates of fMRI opening up to the larger neuroscience community and the other way around, neuroscience community embracing also this imaging, right? I always been one of the of the firm believers in the possibility of complementing, because that's what we're talking about, right? Complementing human neuroimaging with like experimental work in accessible species. And I think the potential it really is enormous. And I think people have just begun to realize, people also especially, that are oriented to human research, of course, and it really branches out in along different directions. We can go from description to explanation, which is really one of the main goals of my own research in my own lab, right? Try to see what we can map in humans and translate that into phenomena that we can explain at the biological level. We can go to signals, like the signals we measure in humans with neuroimaging, to mechanisms, right? From maps to models. And eventually, it really boils down to the possibility in animals to mechanistically probe in a causal way some hypotheses that have been there for a long time, but that we lack the tools to test in humans. And that's I think powerful. And the fact that OHBM has been recognizing this, to me, it's a major step forward for the field. We, as people that have started from the clinical field in animals mostly, we have a lot to learn from human neuroimaging and the approaches that have been developed there. And probably the other way around. I think if we just learn a little bit better what these signals are, what they signify, and what they don't actually reflect as well, I think we can have like a more focused and more effective use of the time also that we spend and the funding we spend in mapping the human brain, which is really what we all care about eventually. So the potential is very high, I think, and very strong.
How does it work? So to someone who, has only human brain imaging experience, which I would imagine is a lot of people who would be listening to this interview, Is scanning a mouse the same as scanning a human? Are there challenges with that? Is it different?
Yeah, clearly there are big differences. Some are technical, some are perhaps more conceptual slash translational. The technical challenges are really daunting, and it's been really difficult, especially for the functional part. Functional is still a little bit of an art, even though now, as the field has been growing, now there are also small community-like efforts that I aim to standardize the method in animals. But basically, just going down to the details. Well, for sure, the most important technical issue we have when we image mice, and because that's what we do. Okay, you can do rats. Rats are already bigger, but mice are very small. So a mouse brain is as big as my, fingernail here. So it's very small, right? 1 centimeter. Uh, the biggest problem is that they are very small. So you need to have like high resolution scanners, so high field basically, okay, and good hardware. Now this is kind of easier than like 10 years ago, 15 years ago. And that's really, probably something obvious, but it's still something you have to deal with on a daily basis, right? Images, getting like high quality fMRI imaging in such a small brain, it's not easy. The second problem, which I think I would say like technical slash conceptual, is brain state. They need to be immobilized, right? Because otherwise they go all over the place. And for many years, we've been using like sedation in the lab to keep them still, which works still great. The problem is that, the conceptual problem here, and this is something that the field will have to grapple with, and it's what is the brain state that the mouse is, right, when you're slightly sedated. And make no mistake, there are also means to go with awake mice. We can do that. We've been doing it in the lab. We've been pioneers there as well. The thing is that mice are always doing stuff, right? They're whisking, they are moving. So this is another big problem that we are dealing with, how to interpret and how to control state. And then there is like clearly the other major issue, which is about translation, right? This is conceptual, if you like. What is worth doing in animals and what is perhaps like not worth doing because we can do it in humans? That's a big question, right? So this is something we've been discussing and addressing, a question we've been addressing in the lab over the past years. I think that a lot of stuff we can do well in mice. For example, my lab has been studying resting state fMRI, and we have a lot of data from our lab, but also other labs, showing that the basics of this phenomenon, that they're really strongly conserved across species. Well, it legitimates us in trying to use the mouse like an accessible species to understand that. And clearly, this has to be applied to every sort of like measure you want to do in neuroimaging. You have to make sure that what you're measuring is really relevant and can be translated. If you want that to have the sort of impact we would like it to have, right? And the one we're discussing tonight. And it's a lot of work and a lot of like controls and sanity checks to make sure that what you're studying is relevant. And but as a field, I think we are getting there. So these are challenges, but there are also ways forward, and I think it's something we can tackle effectively.
It's fascinating. So everything you've described is very interesting. How much of that is going to feature in your keynote lecture? Can you give us a little bit of a preview of what we should expect?
Sure, yes. It's part conceptual also because I would like to take the chance, given that hopefully there's going to be like a big audience, to deliver some concepts that are important, some that are perhaps misinterpreted, especially when you look at the biology of these signals. And it's also a little bit of a research journey. But basically, at the heart of the talk, we want to basically bring forward a simple model that explains fMRI connectivity. People have been using it for many years, decades now. And when we started this work, I mean, in my lab, it was like kind of 10 years ago, I was kind of in shock by, how little we knew about this phenomenon. So, this is something we need to explain, right? What I mean is the rhythms, for example, the brain rhythm that support it, also some simple rules that govern its reconfiguration, for example, that we can use also in brain disorders, when there is more connectivity or less connectivity compared to control. What does that mean? So I think we have like a simple unifying model that I will try to illustrate in the keynote, basically. And this applies both brain disorders when there are alterations. That's why I titled it Disconnectivity. But I also want to cover the biological, the physiological basis of this phenomenon. It is important, I think, also for the use of fMRI to study cognition, plasticity, so in like a healthy condition. And yeah, for the rest, you will see at the keynote.
I'm excited to see the keynote now. You talked a bit about brain disorders. How does that work in mice? What does a mouse with a brain disorder do?
Yeah, that's an important question. I'm glad you asked. That's interesting because it's something I've been putting a lot of thinking into and I have a lot of exposure into because of my initial work in the industry. In the industry, we are also pharmaceutical industry. We were not only interested in drugs, per se, but also drugs as treatment for brain disorders. And there was always a discussion about how good rodent models of disorders are, right? What can you model? What you shouldn't perhaps model in rodents. We've been studying the lab for a few years a complex developmental disorder, which is autism, right? How can you study autism in mice? Well, let me state it clear, and I will state it clearly also the keynote. Mice do not, at least myself, I believe mice do not have autism. But autism, for example, like many other complex disorders, has a very strong genetic component. And genetics can be modeled very well in physiological, or biological accessible species like rodents. And we know we have a lot of data showing that a lot of these genes that are implicated in autism, such as synaptic genes, ion channels, and they are implicated also these genes, for example, in bipolar disorder, schizophrenia. Those are genes that are really evolutionarily conserved, very well conserved. And so what we've been doing, what we as a lab, but also other people, we've been using genetics as a key for a biological deconstruction of the spectrum. And then we can measure connectivity associated with different genetic alterations, and we get interesting results that I don't want to disclose tonight. And by doing that, you can test important hypotheses, right? That in humans is very difficult to test because in humans we can measure genetics, but there are all these other variables such as environment, it's a genetic, variation in the, let's call it like typically developing population that really somehow masks effect, right? In rodents, you can control beautiful environment because, these rodents have their own little mates that are genetically, let's call normal, they don't have mutations. You can control genetic variation because all rodents have the same genetic background, right? And you can manipulate that over development. So you can do a lot of crazy cool things that might have a conceptual impact also in how we interpret, how we, frame and describe these complex disorders. And this will be a message that I would like to deliver in my opinion.
So your work spans a lot of domains. So it sounds like you're working on understanding the basic biological signals, doing things that will have direct application to how things work in the clinic. So it's definitely all very, very exciting and something that we're all looking forward to hearing about in the keynote lecture. Is there anything that you won't describe in your lecture that's happening outside of that? Any other exciting research in your lab that you want to share?
Well, yes, yeah, for sure. I mean, there's a lot more that we've been doing in the lab. One, let's say, promising hot thing is the role of neuromodulatory systems that are important in the brain. Of course, we know, like, for example, neurotransmitters like, I don't know, cholinergic, noradrenergic, many people are beginning to study those, but also like some less studies, for example, peptide systems. There are many, and those are really fascinating because they are secreted by a small cluster of neurons. Like you can count them, right? 100 neurons, and yet they generate this profound reconfiguration of connectivity. That's very exciting, and that's another layer that so far, like in my lab as well, and most neuroimaging research in humans has kind of like set aside. You don't want to account for arousal, and that's neuromodulation. Another declination of neuromodulation in the lab is neurostimulation. We have interesting study using optogenetics when you can produce rhythms, and that's also another exciting approach. again, that allows us to establish causality. By doing that, we can see how rhythms propagate in the brain. And we have fairly preliminary yet interesting results that we should be able to follow up upon soon. That's really cool science that I'm very excited about.
Okay. Thank you for sharing your science. Now, pivot a bit. So you said that OHBM has a lot of young trainees, which we definitely see. Do you have any advice for them, just in general, for trainees who want to, forge their own story to kind of follow a successful path like yours?
Yeah, I guess. I don't know. So, I mean, my case, I think it's important because it's a demonstration that there are many paths for science. So I think when you're young, you shouldn't be too preoccupied about, am I making the right decision? The thing is, have faith in life, and attitude matters a lot, right? And also retrospectively, maybe based on my story, I think an important point that shouldn't be underestimated is really multidisciplinarity. Sometimes for some of my students, they're successful in one thing and they try, to go narrow down. And okay, that's merit in that as well. As I said, there are many paths to science, and this could be one path. But I think people should also try to keep, the options open. Sometimes, and they shouldn't be like scared, right, of what's out there and the complexity. The other thing is, let's remember that we have this big privilege in academia to have this freedom, right? When I was in the industry, I was under like a constrained environment, so to speak, which is company. You have a real boss, and the boss decides what you do, and your career advancement and stuff. Academia is gives you this crazy freedom that if you like it, I mean, if you want it, it's really like it's such a rare privilege, right? However, staying in academia requires hard work, and hard work is fueled, in my experience, only by one thing, which is passion. People are not working hard because, I mean, they shouldn't at least working hard because they are afraid of losing their job or whatever. They should be fueled by passion. So that, once again, that's what first question I ask my prospective students: are you passionate about science? Are you here because this is your passion? So that's really the key important point. Passion first, and then hard work is required, but hard work won't be so hard if you're passionate about what you're doing because time goes by, you don't even realize it, right? And there could be other things. Clearly, and some are obvious. Choose your lab carefully. If you want to be passionate, choose a thing you like, you're passionate about, and then do a survey. Where is this thing pursued? Not all labs are equal. Supervisors, mentors are very important, and you can do a research. You can ask people in the lab, what is this? Do you not follow the flashy things like papers? Okay, that's important. Here you need papers, but that's not the only thing. And that's the other maybe advice. Do not just go for practicality because this place is close to where I live or this place. Do a research because those are formative years you have to make the most. Yeah.
To get to know you a bit more personally, what are you passionate about outside of science?
So yeah, I mean, my biggest passion, like personal level, it's like endurance sport. So I'm a keen cyclist here where we live. There are many mountains, so that's for me like a form of meditation. And ever since the pandemic, I've really tried to carve out some times to do that regularly because it's really clear my head. The other thing I try to do as much as I can, I mean, it's not really about passion, but it's really like the idea to have clear boundaries. I try not to work over weekends unless we have like super tight deadlines to keep the space. And it is hard. This is something that paradoxically, requires discipline, at least to me, because I always have this feeling, oh my God, I should be doing that. I'm betraying my students. I'm letting down my students. I should do this. I should do that. So clear boundaries, and I have this passion for sports endurance, and yeah, that's really my anchor outside of science.
I think this is a great lesson for trainees to know that you can balance work and life and still be a successful person like you. Well, thank you for this wonderful interview. I think this will be very helpful, especially for the young trainees at OHBM, but more generally for everyone. So we're super excited to see what you'll say during the keynote lecture. And thank you again.
Thank you. Thank you, Yohan. It's been a great pleasure, and I'll see you all in Brisbane then.
Welcome to a mini-series of Neurosalience, featuring interviews with some of this year's annual meeting's keynote speakers.